All right, so we discussed the grading, and that is lab 1 has been graded. On the labs, you have 2 attempts to get the assignment graded. There will be the first grading, which will be the due date, and then there will be the second grading, which will be the until date, meaning you can send in the assignment by the until date. And if you do, it'll probably give you a note saying that it's late. It's not late if you can submit it. If it is after the until date, you cannot submit it in Canvas. You will need to email it to me, and then it will be late, and you'll get a late penalty. All right, let me share my screen. Oh, let me remind you that my clock on this computer is a little bit off, and I'm going to be following this clock. And so until I get this clock corrected, we will be starting the class. It's, I think, a few minutes late. And I did mean to trick it into Clark College to get that corrected. And I tried to, it's just that they closed earlier on the day I went in. I think I went in on Friday, and they close at 4 o'clock on Friday or something like that. That might have been in between terms. I don't remember. All right, let me share my screen. Let me show the syllabus, the schedule. So today is October 10th, and we are supposed to be working on chapter 2 and finishing it. Let me remind you, you have quiz 1, which is going to be due this Wednesday. And so you can take this by 11:59 p.m. tonight and till 11:59 p.m. Wednesday. I need to go in and make a few changes in quiz 1, and hopefully I'll do that after the lab. There is a lab today, lab 3, and that's what we'll be doing today. Any question about any of that? All
Is
right.
the quiz going to be on the chapter 110 and the labs 1 and 2 that it says there?
Yeah, it'll be on the start of chapter 2, so not anything we cover today. Chapters 1 and 10, and then lab 0, 1, and 2.
Okay, thank you.
All right. I think I'll leave that. All right, we had talked to here last time on chapter 2, talking about the lipids. And the last lipid we talked about were phospholipids, which are the main lipid in a cell membrane. And when you put phospholipids in water, they will start orienting themselves this way, with the fatty acid tails away from the water and the heads towards the water, and they'll actually start forming a ball or liposome like this. And I don't think anyone guessed, but that should remind you of a cell, because that looks like the membrane of a cell. And then this sort of looks like the shape of a cell. And then the cell is mostly water on the inside, cytoplasm, and then mostly water on the outside. Living organisms are 70% water, but if you think of the blood is mostly water, and a cell in the blood will be mostly facing water on the outside. So the heads face the water on the outside and the water on the inside, and then the fatty acid tails, the tails of the phospholipid, are on the inside of the membrane. Any question about any of that? All right. The third lipid we need to talk about are steroids and sterols. A steroid is a 4 interconnected rigid carbon rings in a structure similar to this, and this is characteristic of the group. If there's an OH attached to the steroid, we call it a sterol. So cholesterol, shown here, has an OH group attached to it. So this is a sterol. And cholesterol is the most common steroid in the human body. Sterols are an important part of animal plasma membranes. Let me blow this up. And there's the plasma membrane, mainly composed of phospholipids, but animal plasma membranes have cholesterol in it. And the cholesterol gives the membrane added strength. A steroid and a sterol is one lipid that is not composed of monomers, meaning this is the sterol, and it's made up of one molecule, which is a fairly large molecule. But my point is, it's not made up of monomers. So not all lipids are composed of monomers, and it's the steroids and the sterols which are not. Any question about any of that? Oh, I should mention also cholesterol is only found in animal products. If you eat only plants or algae or something like that, you will not get cholesterol. So cholesterol is only found in animal products. All right, let's talk about our next important biological molecule, and that are proteins. Proteins are essential in cell structure and function. Proteins may be structural proteins found in bone, muscle, and hair. Proteins may be enzymes which speed up chemical reactions. Proteins can be transport proteins which move chemicals across the cell membrane, or in a eukaryotic cell, they move chemicals or substances across the membrane, like an organelle membrane, as well as the cell membrane. Proteins can be regulatory proteins, which regulate body functions, such as hormones. Proteins can be responsible for movement. I mean, the reason why we move is because our muscle proteins contract, and that's true of bacteria too. The reason why they move is because their flagellin proteins of the flagella are beating. Some proteins are responsible for bacterial toxins. Toxins are substances which are toxic to us. And then some proteins are important as antibodies that help defend the body from foreign microbes that are attacking the body. So as you can see, proteins perform a large number of functions in, well, living organisms, and they are a very diverse group of biological molecules that perform many different functions. Any question about any of that? The main reason why proteins are so diverse is because they are made up of monomers called amino acids, and unlike the carbohydrates or the lipids, there are many monomers that make up the polymers, meaning humans have 20 amino acids, and most living species have 20 amino acids or close to 20 amino acids. Generally, if a species has a different amino acid than we do, they usually throw out an amino acid. This is the generalized shape of an amino acid. It has a central carbon and a hydrogen connected to that central carbon. On one end of the amino acid, there's an amino group, and on the other end, there's a carboxyl group. And then there's a third group called the R group. The R group can be essentially anything, but it's an organic group of atoms that make up the amino acid, and which R group you have will depend on what amino acid you're talking about. So humans have 20 different amino acids. That means that we have 20 different R groups to the amino acids. Any question about any of that? There's one R group there for the amino acid tyrosine. What you do with the amino acids is you simply link them together one after the other to make the protein. All right, there's 20 different amino acids. That means there's 20 different R groups. These amino acids only differ in the R group. And here are some examples of the R groups. Do not memorize them. I'm not going to ask you what is the amino acid glycine or what's the R group of glycine. But glycine, the R group is the simplest. And it's shown in light blue, which is really hard to see right there. But it's simply an H for the glycine. That's the R group. For alanine, it's a CH3 group or a methyl group. For the valine, it's 3 methyl groups. For leucine, it's 4 methyl groups. For isoleucine, it's also 4 methyl groups, just in a different orientation. Some of the R groups are hydrophobic, like this one here. That R group is hydrophobic. So are, I think, most of the ones I showed you previously. Yeah, all of these are hydrophobic R groups. But some of them can have hydrophilic areas to the R group, and we'll call it a polar area to the R group. Like this OH group is polar, and so this R group is both nonpolar, this side, and then polar, this side. This one is mostly polar from the NH here and here, and that one's slightly polar from that NH here, but this group here would be nonpolar, and so is that. All right, any questions about the R groups and the different amino acids? I guess I should mention that amino acids are always polar because the amino group and the carboxyl group are polar. And then some of the R groups have polar parts to it, like this one. Come on. And that one has a large polar group. And then some of the R groups are nonpolar. For a protein, having nonpolar groups on their R groups, they will put these amino acids on the inside of the protein. And then the R groups, which are polar, will be on the outside of the protein if the protein is in water. Now, if the protein is in a lipid, like a protein in the cell membrane, it will put these R group amino acids on the outside of the protein, and then the amino acids, which are polar, will be on the inside of the protein. Any question about any of that? All right, amino acids are linked together one after the other, like links on a chain, and that link is called a peptide bond. What you do is you pull off the OH of this amino acid and the H of that amino acid, and then directly connect that N to this C group. And that bond, which is a covalent bond between the N and the C, is actually called a peptide bond. This bond is actually not only rare in biological molecules, it's actually rare in the universe. And that's why they call it a peptide bond, because although it's rare, it is found in peptides, which of course are proteins. Or I should say a protein is a peptide. A growing protein, meaning a protein which is having amino acids added to it, or a growing protein, is called a peptide. In fact, it's usually called a polypeptide if it's longer than a few amino acids. The difference between the polypeptide chain and the protein is it's only called the protein when the last amino acid is added. Before that time, it's called a polypeptide, unless it's just a few amino acids, in which case it's just called a peptide. Any question about any of that? Oh, I guess I should mention how you get the amino acids to bind is you pull out water here in dehydration synthesis, make the peptide bond there, and then water comes off. When we're talking about proteins, they have 4 different levels to their structure. And I should say they have possibly 4 different levels to their structure. The primary level, the secondary level, the tertiary level, and the quaternary level. The primary level is just the sequence of amino acids, one after the other. That primary level can then start interacting where nearby amino acids will be attracted and repulsed from each other. The secondary level is the interaction only of nearby amino acids. If you have amino acids from very different regions of the protein interacting with each other, that's the tertiary level. And the tertiary level is the supercoiling of the polypeptide into the nearly finished protein. So tying up the protein into a knot, that's the tertiary level. And then some proteins have a quaternary level. It's only the proteins that have more than one polypeptide making up the protein. They have a quaternary level. And the quaternary level is simply when the different polypeptides come together and then start interacting with each other. Any question about any of that? If not, let's watch a little video on the 4 levels of protein structure. Check my volume.
If you're taking anatomy and physiology for the first time, I can guarantee it's not like any course that you've ever taken. And that's why so many students...
In this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid right next to it. The left side, where the nitrogen is located, is the N-terminal of the amino acid. The right side, that has the carboxyl group, is the C-terminal of the amino acid. Now, these two amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the two amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2O. We have one hydrogen left over. We lost two once we lost water. So this is a dipeptide because we have two amino acids combined. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues.
All right, let me back up just a bit there. They're mentioning the hydrogen bonds as the most important interaction between neighboring amino acids, but you can have other interactions, and I'll talk a little bit about that later. You can have ionic interactions. You can have actually van der Waals forces, which are hydrophobic amino acids want to be by another hydrophobic amino acid. And you can have a repulsion, where a hydrophobic amino acid will be repulsed from a hydrophobic amino acid. Any questions about any of that? Okay, so hydrogen bonds are the most important one, which is the one he discussed, but there are other interactions that can happen between neighboring amino acids in the secondary structure.
Acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
As a
All right, any question about any of that? Why did that come up?
In this
twice?
video, we're going to talk about
All right, let me talk more about the 4 levels of protein structure. The primary structure, or the primary level of protein structure, is simply the specific sequence of amino acids in a polypeptide chain, one amino acid after the next. And it is the primary structure which really gives the protein its shape, because the shape will be determined on what the amino acids are, and what they are will determine how the protein will fold in on itself. Anyways, the primary structure is just the sequence of amino acids. Any question about any of that? The secondary structure is the localized interactions between neighboring amino acids. You can actually have 3 different secondary structures. We're only going to talk about 2 of them. The alpha helix, where the protein sort of wraps on itself, sort of like a spiral staircase. But you could have the beta pleated sheet shown here. And the third, which I don't have shown, you don't really need to know, is the random coil, which can be a polypeptide, which randomly coils and wraps on itself. All of the interactions in the secondary structure happen from nearby amino acids interacting with each other. It's the nearby amino acids which will cause the secondary structure to happen as the nearby amino acids start reacting with each other. So these 2 amino acids, it's not shown here, but they're close together and they're forming the alpha helix. It's shown here. So they're interacting with each other in, it looks like, a favorable way, such as forming a hydrogen bond between them. You could also have a favorable interaction, meaning these 2 will be attracted to each other, these 2 amino acids. And it could be simply van der Waals forces, where if you put 2 lipid molecules, or in this case, R groups, which are nonpolar, it'll want to be next to another nonpolar amino acid. And since they're both nonpolar amino acids, at least the R groups are nonpolar, then these 2, come on, mouse. Sorry, my mouse has died. There it goes. Then these 2 amino acids will be next to each other by van der Waals forces. And an example of van der Waals forces is when you put one molecule of lipid of oil in with another molecule of lipid of oil. The oil, all of the different molecules are attracted to each other, and that's why we have a lipid. If the oil molecules were not attracted to each other, they would not form a lipid, they would be a gas. And of course, if they're really strongly attracted to each other, it would be solid. And if you took olive oil and put it in the refrigerator, the oil would solidify and it would be a solid. So that's the way that van der Waals forces can act. And like I said, a perfect example is different molecules of olive oil. They are attracted to each other by van der Waals forces, and that is the nonpolar molecules want to be around other nonpolar molecules. And when they interact with that, that forms the liquid. You have to have attraction between molecules to have a liquid. If you have no attraction, it would be a gas. Let's see. We already talked about hydrogen bonds. So two polar amino acids will be attracted to each other and form a hydrogen bond. You can also have an ionic amino acid. That means one that has a positive charge will be attracted to an amino acid with a negative charge. That will form an ionic bond between them. You can also have repulsion. Like, let's say this amino acid, come on, has a positive charge. That amino acid has a positive charge. These two amino acids will then be repulsed from each other. There is an interaction between the amino acids. So these two amino acids will never come closer than what they are now because they're repulsed from each other. And then you can have repulsion that this one is polar, that one is non-polar. These two amino acids will not want to come together. They're repulsed from each other. Any question about any of that? The point is, the secondary structure happens by the local amino acids interacting with each other. And those interactions can be attraction, such as a hydrogen bond, which is probably the most important bond forming the secondary structure. But it can be other molecules of attraction or other, what do you call it, attractive interactions between two amino acids. And then it can also be repulsion, which the two most obvious are nonpolar versus polar. And then ionic, both positive, they will be repulsed from each other. Any questions about the secondary structure? All right. The tertiary structure is the overall three-dimensional structure of the polypeptide chain, meaning the primary structure will start forming the secondary structure, but then the amino acids will start interacting with each other all over the polypeptide chain. For example, this amino acid here is near the end or the end amino acid, and it's being attracted to an amino acid near the beginning of the polypeptide. And that attraction is not shown here. It's either a hydrogen bond between two polar molecules, but it could be an ionic bond being attraction. And then this one here, it's probably, sorry about that, probably, come on. Let's say this one here and this one here might be being repulsed from each other. The tertiary structure is the overall supercoiling or the super knot of the amino acid to form the finished polypeptide shape. And it could be the protein shape. We'll talk about that in a minute. There is all of the interactions you saw in the secondary structure happening in the three tertiary structure. So you can have hydrogen bonds forming, you can have ionic bonds forming, you can have repulsion between a positive amino acid and another positive amino acid. You can have repulsion between a polar amino acid and a non-polar amino acid. And then you can have an attraction between non-polar amino acids, and that would be a van der Waals forces. We're not going to talk much about van der Waals forces, and I'm not sure, I don't even think your author talks about them, but that's the attraction between two non-polar molecules or atoms. So all of those can happen. And then there's one additional interaction happening in the tertiary structure. and that's the disulfide bonds. That is an attraction between two cysteine molecules. Let me stop this and bring up paint. So we can have an amino acid, a polypeptide that's taking on some shape like this. And it can have the amino acid cysteine here. I'm not going to try and write that, so I'm just going to type it. Yeah, let me see if I can copy that. Nope, I can't get it. Oh, well, just make it over here. And we have another cysteine over here. Oh, come on. So that is a part of the amino acid, meaning it's this amino acid right there. And cysteine has a sulfhydryl group on it. Let me go ahead and put HS right here. I sure wish I could copy that. Oh, actually, I better not copy it. It's going to have the hydrogen in the wrong place. So let me put the sulfhydryl group right here. Well, those hydrogens can come off the sulfur, and then the sulfur can bind to each other in a polar covalent bond. Not a polar, I mean a covalent bond. Go like that. There we go. This makes a link between this amino acid and this amino acid that only happens between the amino acid cysteine. And it forms that sulfur dibridge, meaning two sulfurs connected with a covalent bond. This is a very strong link between these two amino acids, much stronger than the hydrogen bond. And for proof of that, if you get your hair permed, what they do is they put these cysteine sulfur dibridges in to make the perm in your hair. And as you know, that perm will last about a month, even with regular showering. The hydrogen bonds would be simply, my mother, we used to do was put her hair in curls and bobby pins, and then put a cap on and then put a humidifier on, meaning it would put like close to steam in her hair, and then she would take it off, and that simply would put in her hair to give her the curls. Once she took off the curling rolls and the bobby pin, she'd have a curl in her hair. That only forms hydrogen bonds, and the hydrogen bond can break simply with moisture in the air, whereas moisture in the air is not going to break this perm bond. And that's why the perm will last about a month, even with regular showering. Any question about any of that? All right, so the point is that in the tertiary structure, we can also get disulfide bonds forming between cysteine amino acids. And that's a very strong link in the tertiary structure. All right. Many proteins, all they have is the tertiary structure. They don't have any structure higher than the tertiary structure. And that's because the protein is only made up of one polypeptide. That's every enzyme that I can think of, meaning catalase, catecholase, renin, sucrase, all of those enzymes. They're made of only one polypeptide. So the highest level the protein can get is a tertiary structure. But some proteins are made up of more than one polypeptide. These proteins can then form the quaternary structure, and that's the structure of the protein once all of the polypeptides come together and then start interacting with each other. Bonds are basically the same bonds that maintain the tertiary structure. So other weak forces, hydrogen bonds, sulfur dibridges can happen in the quaternary structure. And as you can see, these amino acids to this polypeptide are being attracted to these amino acid of that part of the polypeptide. These amino acids are being attracted to this part of the amino acids of that one. And it looks like these amino acids here are being repulsed from these amino acids. Certain proteins are made up of more than one polypeptide. For example, hemoglobin and antibodies, each of them are made up of 4 polypeptides. And these proteins don't form their full structure, the quaternary structure, until each of the polypeptides comes together and then interacts with the other polypeptides. Any question about any of that? All right, proteins have an overall shape. We can have a globular protein. This protein is roughly spherical. The protein is compact. An example of a globular protein is hemoglobin. You can also have fibrous or thread-like proteins, and these are proteins usually involved in structural proteins of the organism, like collagen or whatever you call it in bone. Anyone take A&P? What is the protein that makes up bone? I used to know that, but I'm afraid for the moment at least, I can't recall what that's made of. No one's taken A&P? All right. Anyways, they make the fibrous proteins. Proteins can also be more complex in that they can combine with other organic or inorganic components. If a protein combines with an organic or inorganic component, we call them a conjugated protein. The conjugated proteins are usually named by their non-protein component, like a glycoprotein is a protein bound to a carbohydrate. Glycogen means sugar, so we're going to say carbohydrate, because usually it's more than one simple sugar joined to the protein. A lipoprotein is a protein bound to a lipid. Any question about conjugated proteins? All right. When we're talking about proteins, it is important for the protein to take on its shape, and it will not have its full optimal function if it's not in its correct shape. And you'll start to lose activity as the protein starts unfolding and taking on less of its correct shape. When the protein starts unfolding, we call it protein denaturation. And denaturation is usually used to discuss a protein that is totally has no function, We call it a denatured protein. It has no function. And that's because the protein has lost its three-dimensional shape and it takes on a different shape, a shape that it's not supposed to be in because it can't function in this shape. How you get a protein to lose its shape is mostly you break the hydrogen bonds, but you can break ionic bonds as well. You can also break van der Waals forces, meaning two nonpolar amino acids which are attracted together. You can break them apart. And then you can have the protein form inappropriate bonds, like form a hydrogen bond where it should not be. There are different ways you can denature a protein. You can put the protein in the incorrect temperature, the incorrect pH, or the incorrect salt concentration. And then that will interfere with the protein's three-dimensional shape, or if it has more than one polypeptide, its four-dimensional shape. Breaking bonds, forming bonds, breaking attractions, forming attractions that shouldn't be there, causing the protein to be denatured. A perfect example of a denatured protein is albumin in egg. When you first crack the egg, you'll note that the egg white, it's not very white and it's translucent, almost clear, and it's a liquid. You can denature that protein by putting it in the incorrect temperature, which we do when we cook our egg, and then the egg white becomes a solid white. That is a denatured protein, or denatured albumin for the egg white. All right, any question about proteins? If not, let's move on to nucleic acids. There are two, DNA and RNA. DNA is also called deoxyribonucleic acid, and RNA ribonucleic acid. Both DNA and RNA consist of monomers called nucleotides. So DNA is made up of nucleotides. Nucleotides themselves consist of three parts: a 5-carbon pentose sugar, shown here, a phosphate group, shown here, and then a nitrogen-containing base, shown here. So that's a nucleotide. There are two purines and two pyrimidines in the nucleotides of DNA. And actually, that's true for the nucleotides of RNA as well. The purines are adenine or A, guanine or G, and you can learn either name, the adenine or the A. The pyrimidines are cytosine, C, or thymine, and thymine, or T. And those are the DNA nucleotides of DNA. Now, how you can know the purines from the pyrimidines is in DNA, the word pyrimidine has a Y in it. The word cytosine and thymine also have Y's in them. Okay, so that's a simple way to remember a pyrimidine from a purine. Adenine and guanine, which are purines, do not have Y in them. Anyways, these are the nucleotides of DNA. RNA has 4 nucleotides. The A, G, and C are all the same. However, RNA does not have the T. they have U or uracil replacing thymine. So once again, there's 4 nucleotides in RNA. And if you remember that uracil replaces thymine, thymine has the Y in it, so it's a pyrimidine. Uracil replaces the thymine. It doesn't have a Y in it, but if you remember it replaces the thymine, then you'll know that uracil is a pyrimidine. The bases of both DNA and RNA combine to each other via hydrogen bonding. And in DNA, we always have A and T binding to each other, or T binds to A, meaning they're complementary bases. And then C always bind to G and G always to C. You never have A binding to C or G because they don't bind. The binding is hydrogen bonds forming between them. Oops, excuse me. Let me blow this up. So A and T have 2 hydrogen bonds forming between the nitrogen of adenine here and here, and then the hydrogen of thymine here and here. Forming hydrogen bonds between them. That is all that's holding DNA together. Each strand of DNA is held by covalent bonds, but the 2 strands are only held together by hydrogen bonds. And the hydrogen bonding occurs between the nitrogen-containing bases, the complementary base pairs. What do I want to say about that? Get that out of the way. Already talked about that. Oh, now I remember what I want to talk about. The sugar phosphate backbone is what holds a single strand of DNA together, meaning we have a sugar here in the turquoise blue color. Come on, mouse. And then a phosphate, sugar, phosphate, sugar, phosphate. So the single strand of DNA is held together by the sugar phosphate backbone. And that sugar phosphate backbone is called the backbone because it's holding the single strand of DNA together. The nucleotide bases do not hold the single strand together. The nucleotide bases are off on the side, forming the steps to DNA's spiral staircase, spiral staircase. And the nucleotide bases hydrogen bond to the corollary nucleotide on the other strand of DNA. So these 2 bases are complementary, we say, because they hydrogen bond, and G and C hydrogen bond to each other 3 times. A and T hydrogen bond to each other 2 times. So DNA is a double-stranded molecule. RNA is only 1 strand. We'll talk about RNA in just a minute. And it's held together by the sugar phosphate covalent bonds holding the single strand of DNA together. The bases will hydrogen bond to the complementary base on the other strand so that the hydrogen bonds are holding the 2 strands of DNA together. Any question about any of that? All right, so RNA is a nucleotide similar to DNA. It's just we have a different sugar. Instead of deoxyribose, we have the sugar ribose. RNA is single-stranded, at least in our cells, it's single-stranded, and DNA is double-stranded. The only time you really see a double-stranded RNA is when we're talking about viruses, and we'll talk about them late in the term. So generally in our cells, unless there's a viral infection, the RNA will always be single-stranded. Once again, the RNA strand is held together by the sugar phosphate backbone and the nucleotides. The bases being A, G, C, and then U replaces T. T in DNA, U in RNA. U standing for uracil. There are three major kinds of RNA. There's tRNA, messenger RNA, and rRNA. When the DNA is transcribing a gene, how does it know to make tRNA instead of making messenger RNA or rRNA? How does the RNA polymerase know which RNA to make? Anyone have an idea? All right, you're not geneticists, so I didn't expect anyone to really get it right. But the point is, the RNA polymerase doesn't need to know. It is simply transcribing the DNA. And if the DNA codes for a protein, that RNA will be transcribed as messenger RNA. But if the gene codes for an rRNA, then it'll be transcribed as rRNA. And if the gene codes for tRNA, the gene will be transcribed as tRNA. So the RNA polymerase doesn't need to know, it just transcribes the DNA, and then it'll make the correct RNA because it's simply copying what is the nucleotides of the DNA. Any question about any of that? All right, each of the three major kinds of RNA are important for protein translation, meaning we have protein translation using tRNA, messenger RNA, and then rRNA. Any question about any of that? All right, let's briefly talk about ATP. ATP is adenosine triphosphate. ATP is the primary energy source for all cellular activities. If the cell needs energy, it most likely will obtain that energy by degrading ATP. ATP is adenine with ribose, that sugar, and then three phosphate groups attached to it, and that we call a triphosphate. In the phosphate groups, you'll note, let me blow this up. The squiggly line indicates that this bond is a high energy bond. And if the cell wants energy, it'll break this bond, and that will release a lot of energy. If the cell wants additional energy, it can break the second phosphate off, because that also is a high energy bond. Any question about any of that? So if the cell wants energy, it just breaks this bond, and then that releases a lot of energy. Oh, come on. Erase that bond. And then I said if it wants more energy, it can even remove that bond. So ATP is made by dehydration synthesis. You simply get ADP, or adenosine diphosphate, and add in inorganic phosphate. You need energy to get this reaction to go, and then that can form ATP, and we have water coming off. You can also break ATP, breaking off the third phosphate group, And then that liberates a lot of energy. So the reaction can go both ways. This will release energy. This will take in energy, but make ATP, storing that energy. So ATP, remember, it's the cell's energy currency. Whenever the cell needs energy, it usually gets it from ATP. All right, what molecules have we discussed that have hydrogen bonds in them? And these hydrogen bonds give the molecule an important chemical property. Come on. Somebody's got to know. Let me check and see there's still people there. Yeah, there's still people here. What molecules have we discussed today that have hydrogen bonds in them? Well, we're going to wait here for 14 more minutes. Well, think about that. And then you can also think about what would happen to these molecules if all the hydrogen bonds were to disappear. Unless somebody's going to contribute, I guess I'll end the lesson.
Water has hydrogen bonds.
Yes, water has hydrogen bonds, and it's very important for holding water together. Otherwise, you wouldn't have liquid water. And then the same with DNA. It has held together by hydrogen bonds. Just checking to make sure we're on schedule. We are. And what would happen if all those hydrogen bonds in water and DNA were to break? Well, in water, would we have liquid water if all the hydrogen bonds were to break? Okay, think about that. And obviously, if all the hydrogen bonds were to break in DNA, we wouldn't have double-strand DNA. If you don't have double-strand DNA, it would be difficult to, well, transcribe the DNA and translate the DNA, or not translate, copy the DNA. Meaning we do need double-strand DNA. All right, if there's no questions, I'm going to end it here, and I'll see you at 6:30 for the lab.
Thank you.
Bye.